Hello, everybody. Thanks for joining this webinar today. We're very pleased to see so many of you joining. We have participants from 5 continents, from many different backgrounds, including academia, policymakers, journalists, NGOs, experts, and also citizens. So this is, of course, a challenge also for our speakers to give speeches that can be attractive to all of you and where everybody can take some takeaways with them. But in any case, we're very happy that so many of you were interested to join for the topic today, artificial intelligence and genetic engineering, a biosafety issue. So we decided to focus on the biosafety aspects today because this debate on the convergence between artificial intelligence and genetic engineering often centers around biosecurity aspects, meaning that, for example, malicious actors could use the tools at disposal to engineer biology and generate new pathogens, viruses, toxins, or other outputs that could be weaponized. However, possible biosafety issues often fall underneath the radar. These include applications in plants and animals that may, even so well-intentioned, result in substantial risks for nature and the environment. My name is Astrid Österreicher. I'm a policy officer for Testbiotech based in Brussels, dealing mainly with European Union policy process on genetic engineering. As you know, we have a policy discussion ongoing at this very moment that we are speaking. So this will also be one of the focus points of our webinar today. And without further ado, I would like to now hand on the word to Mrs. Franziska Achterberg, who is an expert from SOS Save Our Seeds, and who will give us an introduction into the topic and a good overview to start the discussion. Please, Franziska, you go ahead.
Thank you, Astrid. Welcome, everyone. I am hoping to share my screen now. Astrid, is that okay? Is that happening?
Yes, it's perfect. Thank you.
Perfect. Welcome, everyone. So I'm Franziska Achterberg. I work at Save Our Seeds. We are a Berlin-based initiative working for robust regulation of genetic engineering. Our focus is on agriculture. In fact, we see genetic engineering and the release of genetically engineered organisms into the environment as a dangerous distraction from ecological and organic farming innovation. I am going to hopefully go through with my presentation, but I don't see it works. Hang on. Yes, I'm going to talk about those latest developments in genetic engineering that we are observing from where we sit. And that includes, obviously gene editing and then the use of generative AI tools. And most of what I'll be saying is based on a report that we published in January. So I will have a few slides on those latest trends in genetic engineering and then come also to the legislative process that is going through in the EU right now, which would result, if it gets finalized, in the deregulation of certain plants engineered with new GM techniques, such as CRISPR-Cas, including, of course, the plants that are engineered with the use of generative AI. So I don't think I need to say much more about gene editing, right? So this latest tool in genetic engineering, discovered in 2012, earned a Nobel Prize for its 2 inventors. The use of generative AI, which we see as transformative to gene editing and genetic engineering more widely, is even more recent, and it really continues to accelerate. So I will talk a little bit about what that is and what that means. So developers have used the AI architectures that are used in chatbots, such as ChatGPT, or in image generators like DALL-E, and trained them on the languages of biology. So that would include protein and genome sequences, which have become available at a massive scale recently. And that really is the fuel. That is really the basis, the resource that all those tools depend on. So we have a massive wealth of data on DNR and RNA sequences, on proteins, on metabolites of plants, etc., which has really been exploding over the last couple of years, and it will continue, obviously, to increase. When it comes to those AI tools, they are both descriptive and generative. So we have tools that analyze this biological data and can make predictions. What will happen when you change this, when you change that? How is that going to influence your organism? And then you have tools that are generative and that can literally design functional DNA, RNA, and protein sequences, including sequences that are entirely new to nature, that don't exist in nature. We distinguish different types of tools on the basis of the type of data that they're trained on. So you have the basic, research assistants that are trained on scientific literature. But then you have these protein models and DNA models. And as I said, I'm going to speak mostly to plants, so I'm not mentioning the RNA models. We have models based on protein sequences that analyze these proteins and simulate what happens when you make certain changes. The most famous of those is AlphaFold, the model from Google that earned Demis Hassabis and two of his colleagues the Nobel Prize in Chemistry last year. And then you have 4 models trained on plant DNA. The biggest of those is called AgroNT, and it is trained on 48 plant species. And then you have multimodal models, which combine the different types of biological sequences and even natural language. So the latest of these, believe it or not, is called chatNT. It comes from InstaDeep, and InstaDeep says it is a multilingual conversational agent that fuses biological sequences and natural language, of course English, into a shared vocabulary. So these models, of course, widen the range of people that can engage in genetic analysis, but also genetic engineering, because you can use these tools much more easily. I will give you one example, which we also outline in our report. This is about poplar trees that have been engineered to lower their lignin content. This is supposedly better for paper production. So there are 21 genes involved in lignin synthesis in poplars, and there are over 69,000 potential editing strategies to lower that lignin content. And so this US company, which is a spin-off from a public university in North Carolina called Treeco, they have their own AI tool, proprietary tool, which can simulate these strategies and predict the impact on other important parameters such as wood composition, etc., and can help the company to identify the best possible strategies. So in the end, the company only tested in the lab 7 of these gene editing combinations, and it claims that it's been able to decrease lignin by 50%. So the important thing here is the sort of change in workflow. So first it goes into what they call the dry lab, which is your computer model, where these best strategies are identified, and then it goes only into the wet lab, where only a few strategies will be tested, and then of course field tested if they're successful. So in our report, we've looked at both. We've looked at what are the opportunities that the use of AI brings, and in particular in conjunction with CRISPR-Cas. So we see that it is used to optimize and the tool and the use of the tool. So like in the example that I just mentioned, it is used to identify the target sequences and the best possible editing strategies, but there are also ways now to improve the tool as such. And importantly, and we're going to go into that more in detail later, we have seen that with the use of AI, the capabilities of CRISPR-Cas gene editing have been expanded. So we're going beyond what famously George Church called the blunt X, CRISPR, the blunt X, the DNA vandalism, you know, just cutting somewhere and leaving a lot of damage behind. Now it is possible, supposedly, to also modify gene expression, so do a lot more, you know, specific and targeted tweaks to genomes that arguably couldn't have been done without the use of these models. Now, we've also looked at possible downsides. If you consider that gene editing by itself is imperfect and has unwanted effects, you know, you add the unwanted effects of imperfect AI, you know, you get the perfect mix here, perfect explosive mix, you know, whatever can go wrong. So AI is known, you know, to be only as good as the data it's fed with, you know, it's trained on. So you get distortions, you get mistakes that are simply introduced via the data. You all know from using ChatGPT that AI chatbots hallucinate like hell. So it is to be expected that that is not just ChatGPT, but potentially also these models used in genetic engineering. And importantly, you have this effect that you don't quite know what your tool has done. So you put something in, you get something out, but in the middle, it really is a black box. You don't actually know why a certain decision, a certain strategy was chosen. So all of that, as I said, is in our report of January. Just really these thoughts, because as we will see, you don't really have experimental data to say how well these algorithms work. 2 slides, as I said, on EU GMO regulation, just to set the scene here for further discussion. So today's regulatory system in the EU requires for each GMO an EU authorization after a risk assessment that is carried out centrally, mostly, by the European Food Safety Authority. The applicants need to submit a detection method. There is a requirement for consumer labeling, so consumers need to know whether or not the food or whatever product they buy contains GMOs. There is a traceability requirement throughout the supply chain. Importantly, there is not just environmental risk assessment, but there is also environmental monitoring when it comes to cultivation to spot anything going wrong, you know, post-release, post-marketing. And in the EU, countries are able, and that is only a new law from 2015, to ban the cultivation of EU-authorized GMOs. So that is the baseline. At this moment, there is only one GMO authorized for cultivation and used in very, very small quantities. And I'm not talking about imports. Now, a proposal has been on the table since July 2023, so 2 years, which aims to deregulate certain GMOs, and that is GM plants that have been engineered with gene editing and similar technologies. The EU calls those technologies new genomic techniques. So this is not genetic modification techniques, but in fact it is. And they're the more recent ones since 2001. The objective of that legislation is to facilitate market access and to circumvent public opposition in the EU. So the most important element of that proposal is what is called the category 1 in plants derived from new genomic techniques, category 1 NGTs, which are defined as not having more than 20 genetic modifications. There are some more specific criteria attached to that. The important thing is that the EU, under this proposal, would regard those plants, GM plants, as equivalent to conventional plants. And hence, almost all GMO requirements that I've mentioned, including risk assessment, including monitoring, would not apply. The only thing we would have in terms of labeling is the labeling of the plant reproductive material, aka seeds, as category 1 NGT. So here are this is my last slide, a few observations. So in this context of deregulation, you can imagine that the criteria for these category 1 NGT plants, which practically fall out of all EU GMO requirements, in a sense will be used to define the design room for AI-based GM plant design, right? If you're a company, you want to bring something to the EU market, you will ask your tool to design a plant that sticks to those criteria and that will not need a proper EU authorization, risk assessment, etc., etc. Arguably, you will have an enhanced speed and capabilities here with AI, but the shortcomings of AI also apply. As I said, no experimental data so far to actually see how well that works. And something that you might have noticed already, there are some new players that are entering into plant genetic engineering. If you look at who's the owner of these tools, you get Google, you get InstaDeep, you get completely new companies that have not so far been involved. So there is a real shake-up of the scene. And with that, I hand over back to Astrid.
Yes, thank you very much, Franziska, for this, I believe, very condensed but also very concise introduction into the topic. And so, as you mentioned, just to try and sum it up, so according to estimates, more than 90% of gene-edited plants currently in the pipeline would fall into this category 1, which you described. So this means they would go without risk assessment. And now with the help of AI, it would become even easier to, develop plants that would then fall into this category where there would be no more risk assessment, whilst, of course, these plants can still have risks for the environment. And I think this is also the crucial point which will be illustrated by our next presentation by Mr. Juhas, a researcher at Testbiotech. If you could tell us about this experiment that you have carried out to show how easily these criteria proposed by the European Commission can be circumvented. Please go ahead.
Yes, thank you, Astrid. Okay, in the first talk, Franziska gave an overview about the status quo of the current developments in the field of the convergence of artificial intelligence and genetic engineering, while in my talk, I'm now coming really close to the title of the report, When Chatbots Breed New Plant Varieties, in terms of we've performed a small-scale experiment to answer the question if it's really possible to design the genetic blueprint of an insecticidal NGT1 maize with a publicly available AI chatbot. But before we go into detail, I would first like to show you the workflow of how we arrived at the design of the insecticidal NGT1 plant and what specific concepts we actually pursued during the development process. In the beginning, it was actually Bernd Rodekow from Aurelia Foundation, which are engaged in bee protection, who had the idea of simply giving a publicly available AI chatbot a try to design such an insecticidal NGT1 plant. In these initial trials, the AI had good ideas by creating quite interesting scenarios for obtaining such plants, but when we had a closer look into the details, they were often imprecise or exact details were missing. That's why we set out to start a small-scale experiment to design the genetic blueprint of an insecticidal NGT1 maize that is potentially toxic to Lepidoptera using the ChatGPT 4o Deep Research agent with the requirements or the design room for so-called Category 1 NGT plants proposed by the EU Commission as a guideline. First, the chatbot offered several strategies for the design of insecticidal NGT1 plants. Second, from these strategies, we followed a plausible design to exchange the promoter, also called promoter swap, of a serine protease inhibitor, SPI. And third, as a further approach, the chatbot realized the design for the inactivation of repressive cis-regulatory elements of this protease inhibitor. Here you can see an overview of the technical steps to achieve the design of the insecticidal NGT plants, starting with an initial prompt of the basic idea. The chatbot provided several scenarios and offered various options for realization, which I will show you later. We asked for detailed sequence information for which the chatbot provided the corresponding specifications, resulting in a design for an in silico NGT plant that contains a higher concentration of a toxic substance, where insects feeding on it may absorb large quantities of this toxic substance and die. So how did we get there? We started with the following prompt. I want to develop an insecticidal NGT plant according to the NGT1 criteria in Annex 1 in EU Proposal 2023-411 and recited the criteria of Annex 1. The strategy may be aimed at modifying cis-regulatory elements, but if there are more effective strategies, I'm also interested in these. The insecticidal NGT1 plant should at best be a crop plant, but if the strategy works better with a model plant, such as Arabidopsis thaliana, that's fine too. ChatGPT wanted to narrow the question by asking for special target insects and plant species, which we randomly answered Lepidoptera and maize, leading the chatbot to research strategies to develop such an insecticidal maize. After the specification, the chatbot delivered various target genes and options for genetic engineering interventions, from which we've randomly chosen the following two, simply because they were the first on the list. First, a cysteine protease that digests the peritrophic matrix in the larvae gut and weakens digestion and defense of the insects. And second, a serine protease inhibitor that inhibits proteolytic digestion enzymes, resulting in reduced nutrient uptake. Several intervention strategies were also suggested, including native promoters to be exchanged by stronger or constitutive cisgenic promoters, so not transgenic but already present in the breeder's gene pool, or the removal of binding sites for repressive regulatory elements. Unfortunately, the approach with the cysteine protease was not successful because even after 6 attempts, the chatbot did not manage to find the right gene with the right ID in the database. Moreover, it was not able to correctly distinguish between proteases and kinases. We therefore skipped the cysteine protease and moved on to the serine protease inhibitor SPI, with which it was possible to successfully design the genetic blueprint for the promoter swap, shown here as option A, and the inactivation of repressive cis-regulatory elements, shown in B. We first started with the promoter swap of the serine protease inhibitor, shown on the right, where you can see the coding region of the SPI gene in yellow and the endogenous infestation-induced promoter to be replaced by cisgenic, constitutive, or stronger promoter. As a small reminder, promoters are DNA elements that are located upstream or in front of the actual coding region of a gene and are essential for the initiation of transcription and therefore also responsible for the expression rate of this gene. We prompted the chatbot to create a guide RNA design and editing strategy for an insecticidal NGT1 maize plant that is toxic to Lepidoptera, where SPI shows a permanently high expression pattern in above-ground parts of the plant, by replacing the endogenous SPI promoter with either a constitutive promoter or a stronger wound-inducible promoter, where both promoters have been proposed by the chatbot previously. The NGT1 maize plant must meet the requirements of the EU Commission Proposal 2023 Annex 1, and relevant publications are the following. The chatbot answered the prompt by providing detailed sequence and target information, first for the guide RNAs that are used for guiding the CRISPR-Cas nuclease to the desired target sequence to cut out the endogenous promoter. On top, ChatGPT offered detailed information for the guide RNA upstream of the SPI promoter, covering the gRNA sequence including PAM, the target nucleotide position on chromosome 6 in the maize reference genome B73 deposited at NCBI database, the approximate location 2 kilobase pairs upstream of SPI coding region, and information concerning possible off-target effects. Below, the same for the downstream guide RNA, leading to the cutout of the endogenous promoter to be replaced. Second, as the chatbot chose the promoter swap to be performed via homology-directed repair by providing a donor template that consists of a left arm in between the exchange promoter and the right arm, the correct sequence and position of the donor template were provided, both for a promoter that mediates constitutive gene expression in all tissues and developmental stages, as well as for a stronger promoter that responds rapidly to wounding in leaves but not in seeds or pollen, again both corresponding to the reference genome data. After the successful promoter swap, the second approach we were following was the inactivation of repressive cis-regulatory elements of SPI, shown here as option B. Cis-regulatory in most cases means elements upstream of the coding region of a gene, like for example promoters or binding sites for either enhancing or repressive elements, like transcription factors that for example prevent or lower the transcription rate of a gene. So we prompted the chatbot to identify known cis-regulatory elements of SPI in maize and to propose point mutations using CRISPR-Cas that knock out negative regulatory elements and subsequently lead to significantly stronger SPI expression. Suggestions on the principle were given by reference to a review paper. From the cis-regulatory elements suggested by the chatbot, we made the following selection. We took the RAF1 motif, the worky box, and an upstream open reading frame start codon, where the first two are binding sites for repressive transcription factors, and the upstream open reading frame inhibits translation initiation through ribosome stalling at the SPI mRNA. We selected these three elements because their knockout should result in less repression or better said an increase in SPI gene expression. The effects of inactivation should at best accumulate, and since the chatbot offered a base editing approach for this scenario, the proposed interventions should utilize the same type of base editor, in our case an adenine base editor that converts adenines to guanines, leading to an inactivation of the binding sites. So the prompt was to design a strategy for the production of an insecticidal NGT maize in compliance with NGT1 criteria in Annex 1, in which a few point mutations in the three above-mentioned cis-regulatory elements eliminate their repressive effect on SPI gene expression. Please provide detailed sequence information on the repressive cis-regulatory elements to be edited, the point mutations, and the gRNAs. Also for this scenario, the chatbot again provided accurate information, which I will show you exemplarily for the worky box, representing number 2 in our approach, repressor number 2, where detailed information for a suitable motif to be edited with correct genome position, appropriate guide RNA sequence, the base editor type, the optimal editing window, and the expected mutations of the adenine to guanine is provided. So after a week of playing with AI, we actually managed to realize a design for an in silico insecticidal NGT1 plant, exploiting the design room of Annex 1 of the Commission proposal that has all the detailed information correct and matches the publicly available genome databases. However, what no longer needs to be investigated due to the absence of mandatory risk assessment for NGT1 plants is what are the effects of the changes in toxicity on non-target insects that, for example, feed on the pollen or on soil organisms and their interactions with the plants. Are there unintended effects in plant composition and metabolism that may affect food safety or its nutritional characteristics? What are the consequences for the food webs and ecosystems? And what about the risks of gene flow and the development of resistance in pest insects? So while it is often discussed that AI may be abused for bad purposes, causing major biosecurity risks, like for example bad actors develop dangerous viruses, our example demonstrates risks to biosafety, meaning risks for the environment and biodiversity. And it shows that more attention has to be paid to the protection of biodiversity and food production. The significance and the use of AI in designing or developing NGT plants and other organisms is evident and already taking place or gaining momentum, but has nevertheless not been taken into account in the EU Commission proposal, making it already outdated before it has even been implemented. And since several companies are already using AI to develop NGT plants, it has to be assumed that their algorithms are much more effective than publicly available versions of ChatGPT. And that's why our small-scale experiment does not stand in empty space, since there are already several AI tools available that are specifically developed for plant breeding, either by academia with pipelines and platforms for redesigning cis-regulatory elements in plants, and tools for identification and editing of upstream open reading frames in plants, or by companies using their own proprietary AI tools like FloraBird from Inari, Genix Traits has an AI for upstream open reading frame editing, or Phytoform using Creative for cis-regulatory element design. Finally, I would just like to draw your attention to a few publications on the topic of the convergence of AI and genetic engineering, which you can find on our homepage or that of Save Our Seeds. And with this, I'd like to thank you for your attention and look forward to the discussion.
Thank you again, Matthias. And then we would now move on to Dr. Christoph Then, who is the Executive Director of Testbiotech. And he has prepared a presentation on the technical tuning of plants and animals via new genomic techniques and artificial intelligence. And basically this is about organisms where we don't see new genes inserted, but where we just have some changes at the regulatory level. But Christoph, you will go into this and explain in detail. Please, the floor is yours.
So thank you very much for your interest, and I asked my colleague to share the slides, and currently, yes, this looks perfect. Thank you very much. So I'm shortly trying to introduce the issue of interfering gene regulation via new genomic techniques and artificial intelligence. And what you see below is a patent application on exactly the topic I'm talking about. So it's about how to identify genomic sequences which are involved in gene regulation, and how to then by generative AI to change it, and then to implement the change via CRISPR-Cas nucleases into plants. So this is far away from being futures. This is already a matter of patent applications and being used widely by companies, as already was explained by other speakers. And as far as I understand, this is a private AI program which will not deliver data to any other company or to the public. Next slide. So to frame the issue, we see a few technical strategies for producing NGT1 plants. One could be to introduce changes in DNA for new or chimeric proteins, but we do not see very much cases in this regard. Maybe by unintentional changes, new proteins will emerge, but there is not really much targeted interference with the plant genome in this area. What we see a lot is deletion of functional genes for undesirable proteins, for example, to lower gluten content in wheat. There are several cases to delete just the function of a gene for producing specific proteins. But what we see the most currently is exactly to interfere with gene regulation, also like we did with the maize, to change the expression of endogenous plant genes. And this has a good reason, next slide, why many companies go this way. Interfering with gene expression sounds maybe too derived to small effects, but this is not the case in biology. It's a really powerful instrument if you consider that the organs within an organism, such as humans, are all stemming from the same genome, and the only difference between the organisms is the way how the genes get expressed. Then you get a better understanding what gets possible if you understand systematically to interfere with gene regulation on the level of the genome and the cells. Next slide. And also evolutionary traits like the extremities in whales are largely governed by gene expression, which is made visible by this picture, by this figure. So we see in gene regulation that it also shapes forms of life, but we know that there are, of course, endlessly possibilities for new life forms to occur, but in the end, only very specific life forms end up in reality, get materialized. Next slide. So what the researchers talk about, or the development biologists talk about, is a developmental bias in evolution, which also then is relevant for plant breeding, which is the topic of this workshop. So the potential, as I said, for various life forms is huge, but certain phenotypes become materialized, like whales do not have gills. And underlying mechanisms for this developmental bias, these constraints, can be described or explained on the level of the genome, the cells, the organisms, and the population. And there is where the great attraction, the great promise comes from for CRISPR-Cas nucleases. Next slide. Because these constraints are also affecting plant breeding. They are limiting the results in classical breeding. Also, if you would consider random mutagenesis, there are clear limitations in the outcome of these techniques. But the new CRISPR-Cas machinery, next slide, allows what also was called a crack in creation. These nucleases can overcome these constraints, or many of these constraints, and it's now possible to intervene with gene regulation and the characteristics of the species to a much higher degree than ever before. Next slide. So what is basically new about new genetic engineering? These NGTs can overcome many constraints posed by evolution. The interfering with gene regulation becomes one of the most powerful and most frequently applied approach in producing NGT plants and also animals. And in the following, I show you some examples to exemplify that a wide range of characteristics can be introduced into organisms that are absent in the existing species, but without introduction of additional genes. And by doing so, NGTs can go far beyond what was possible even by previous techniques, transgenic techniques, or genetic engineering. Next slide. So one of the examples, I first start with some animal examples, go shortly through it. Most well-known example is maybe the dire wolf, which was developed by company Colossal Biosciences. which has created a genetically engineered wolf who's been altered in 20 target sites without inserting additional genes, and basically by interfering with gene regulation, this wolf became stronger, larger than a modified coat, and it was even claimed to be similar to an extincted species known as the dire wolf, but in fact, of course, it's just a genetically engineered gray wolf, but it shows how much power interference with gene regulation can, how much effects it can render on the phenotype of the specific organisms. Next slide. Another example from the same company is the woolly mammoth mouse, which has the following background. The company Colossal Biosciences identified so-called mammoth genes that they want to introduce into the genome of elephants to what they call resurrect the woolly mammoth. To test the genetic effects, mice first now were, because it's easier to do so, were manipulated to show longer and curly hair like mammoths. And again, most of these effects were just caused by interfering with the gene regulation of the indigenous genes of the mice. Next slide. And this is an example for a fish which is already in the market in Japan. In this case, the CRISPR-Cas technology was used to block gene functions which repress the growth of muscles in the red zebra, and this fish consequently shows higher muscle mass, a larger body, a reduction in body length, and also abnormal position of its vertebrates. So again, this example shows that interference with gene regulation can have far-reaching effects. So now we come to plants, also 3 examples. Next slide. One of them you already know, it's the insecticidal NGT maize. It was designed with the help of artificial intelligence, as just explained, and basically it's also an experiment which deals with gene regulation, with the concentration, with the expression of endogenous protein, and maize with similar properties was already developed before with means of transgenesis, with the old genetic engineering tools. And of course, this plant would have to undergo environmental risk assessment, that's very clear. But according to the existing proposal of the European Commission, this NGT plant, this NGT maize, would not require mandatory environmental risk assessment. So we have double standards for the same traits. Next slide. So another example, which is already on the market now, it's not driven by artificial intelligence, but just by NGT, and interfering with gene regulation, is the GABA tomato. The tomato shows an extremely high level of this GABA, which was just achieved by interfering with the plant's own genes again. And the concentration of GABA is supposed to be so high that consuming the tomatoes is supposed to relax the blood pressure and lower blood pressure, to relax, and also to improve sleeping at night. So again, according to the existing proposals of the European Commission, this NGT plant would not require mandatory environmental risk assessment, despite its new to nature. Next slide. And this example is now closer to wild plants, because these also would be deregulated. And in this case, it's a poplar tree where gene regulation was changed in a way that the poplar first flowers already after 4 months, or in nature, this species does not flower before 7 or 10 years. And according to the existing proposal, again, also this poplar tree would not require environmental risk assessment. So now let's go to the next slide, and I'll come to the role of artificial intelligence now in shaping gene regulation. And here we see specific programs that were developed to generate new variants of DNA that interfere with gene regulation. So we have, we'll basically shortly introduce 3 examples, one for plants, one for humans and animals, and one for microbes. Next slide. So this was already introduced. It's the artificial intelligence program called Florabert, also used by Inari. Next slide. And this was also published in 2022. And this artificial intelligence learns from the regulatory motifs from promoters in nearly every publicly available sequenced plant's genome. So this is a huge amount of data that can be used by this program. And of course, it's easy for the program also then to generate new variants of these promoters. Next slide. This is for humans and also for mammals. It's called malinois. In the peer review, in the preprint of this publication, you find the following summary: that this program can be used to engineer regulatory elements and write new regulatory codes fit for purpose for all across vertebrates. Next slide. And similarly, on microorganisms, we have a tool interfering with gene regulation. Here it's a collection of nucleate language models to generate species-specific functional promoters across dozens of species within microbes. So all these three examples show the power of artificial intelligence converging with NGT and SynBio, and in the end resulting in a new dimension of possibilities, of promises, but also of risks. So let's come to the end of my presentation. When we now come to the next slide, I will shortly talk again about the planned EU regulation. And here we see that the European Commission would propose a regulatory system that would exempt more than 90% of the currently known NGT plants from mandatory risk assessment, and it's based on what could be called a magic threshold of 20 genetic changes that are declared to be as safe as conventional breeding. And there is another proposal by Parliament which mostly focuses on new proteins or chimeric proteins. And this proposal maybe then would not consider even the interference with the regulatory sequences as just explained in my presentation. So this would only regulate then very few cases if this would be relevant at all. Next slide. So I think we should agree on minimum standards for NGTs. Every organism should be tested for intended and unintended genetic changes. The releases should be controllable in terms of space and time. Transparency, traceability, monitoring, and labeling must be guaranteed throughout the entire production chain. And patents, which is not now in the focus of my presentation, but which is a major topic in the negotiations, should be restricted to the processes of genetic engineering. And beyond that, we should discuss, next slide, how to contain the convergence risks of artificial intelligence and NGTs. There are experts in the field which know much better than me about these risks. And we think these risks are relevant for biosecurity as well as for biosafety, as we also try to show with the example of the NGT maize. And this is not about regulation of specific cases. This is about regulation or containing the whole technology, for example, by promote independent risk research. I think there was already one question here. In European Union, we do not find a lot of independent risk research. Most of the research is driven by the applicants which want to develop new organisms obtained from new genomic techniques. Another demand would be to expand state capacities to protect people and the environment, and also to introduce prospective technology assessment, which again is not dealing with single cases, but with the overall impact of these organisms onto the environment. And of course, in current situation, political situation, these demands or these proposals do not have a strong lobby. most about the ideas coming up now in European Union, but also of course in the US, is more about deregulation. But in the end, I think there is no alternative than to try to really develop such a containment strategy for this technology if we want to protect our own living and our own livelihood and the environment as necessary. We will not escape from the problems just by hiding away and proposing future regulations of NGTs, which basically is a deregulation. This will not work. So thank you very much for your attention. And my last slide is very, how do you say, no picture, not a big message, just a short summary. Thank you very much.
Yes, thank you, Christoph. And of course, the question that needs to be asked is also in a situation where biodiversity is already under huge threat from all kind of different sources, should we really release without any risk assessment plants from this new technology, given all the risks for environment and health? Would this really help biodiversity, or would it be an additional burden on our ecosystems? Thanks again, Christoph, for the presentation and also for the different examples of what can be achieved with new genetic engineering today. If we think back some 20 years ago to the products we saw back then developed with transgenesis, I think it's actually quite impressive to see all that is possible today. As said before, genetic changes can now take place faster and more comprehensively. And that's why we actually need more regulation, not less. Our next speaker will be Dr. Juliane Mundorf from the German Federal Agency for Nature Conservation. She's a researcher and the co-author of a publication which is already published as a preprint and which is called the European Commission's Regulatory Proposal on New Genomic Techniques in Plants, a spotlight on equivalence, complexity, and artificial intelligence. We're very glad to have you here today, Juliane. You have the word.
Good afternoon, everyone, and thank you very much for the kind introduction. We already had some interesting talks today on the upcoming convergence of genetic engineering and AI. And I would like to delve even deeper with you into the interplay between development and the regulation of GMOs. For this, I would like to present you our recent publication entitled the European Commission's Regulatory Proposal on New Genomic Techniques in Plants, a spotlight on equivalence, complexity, and artificial intelligence, which was co-authored by Samson Simon and Margret Engelhardt from the German Federal Agency for Nature Conservation. So we published this policy brief a few weeks ago in June as a preprint, which you can find also online following this QR code. The objective of the current European regulation for the deliberate release of GMOs is to protect the human health and the environment. So since its adoption, biotechnology has developed in great pace, also with regard to genome editing methods, which are also called new genomic techniques. And in recognition of these technical innovations, the European Commission proposed 2 years ago a lex specialis to adapt the current European GMO regulation. So the aim is to separately regulate certain GM plants generated with NGTs and enable a simplified, merely technical verification process without risk assessment. In the proposal, it's suggested to consider NGT-generated plants as equivalent if the genetic modifications could also occur naturally or be produced by conventional breeding methods. So the definition of equivalence forms the foundation of the entire proposal, from which all other assumptions are derived, including the risk potential. So let's have a look at how the regulation of GMOs works in the EU. The current GMO regulation applies to all kinds of organisms that have been genetically modified using all kinds of genetic engineering techniques, including NGTs. It includes a comprehensive case-by-case risk assessment. The COMP's proposal, or more specifically Annex 1, concerns only plants that have been modified with NGTs. Here, GM plants should be assigned to two different categories if they fulfill specified criteria. Fulfilling the criteria of Annex 1 would qualify a GM plant as an NGT1 plant. Such plants can have up to 20 genetic modifications at targeted sites in the genome, and possible genetic modifications could be insertions or substitutions of up to 20 nucleotides. Deletions and inversions can have any size. It includes cisgenesis with sequences from crossable species as long as no endogenous genes are disrupted. And on top, any other modifications existing in the breeder's gene pool. So assuming that the risk of such NGT1 plants is proportional to that of conventionally bred plants, it is proposed to skip risk assessment. NGT2 plants are simply defined as a NGT plant other than a category 1 NGT plant, which can be interpreted to cover also intergenesis. For such NGT2 plants, an adapted, possibly reduced risk assessment is proposed. I would like to draw your attention to the two threshold criteria. So these represent molecular criteria considering only the DNA level, and as a consequence, neither the genetic context or the functionality or any phenotypic or risk-related outcome is considered. In our policy brief, we concentrate on these two threshold criteria and their scientific basis. So we look at genetic changes in the light of statistical probability, genetic context, and combinability of mutations. And afterwards, we dive into the technical innovations of NGTs that make genetic precision design possible. In addition, we take Annex 1 as an implementation framework for future NGT1 compatible plants. So we dissect the impact of AI on the development of potential NGT1 plants, and we shed light on the underlying complexity and risks of potential NGT1 plants. So the objectives are to decipher whether the assumption of equivalence is scientifically justified, a valid argument for suspending risk assessment, future-proof, and ultimately suitable for regulatory purposes. So here comes the first criterion. A NGT plant is considered equivalent to conventional plants when it differs from the recipient parental plant by no more than 20 genetic modifications in any DNA sequence sharing sequence similarity with the targeted site. So this criterion is according to the related technical paper based on general mutation rates, which considers all genetic changes as equally likely. However, there are three other aspects that I would like to address on the next slides: probability, context, and combinability. So first of all, even if every generation mutations may happen, how likely is it that up to 20 mutations occur? Desired mutations. To illustrate this statistically, I would like to use a simplified lottery example with you. A general mutation rate would be comparable to the chance that any numbers are drawn in the lottery every week, which is 100%. In contrast, the likelihood that the numbers you actually selected are drawn is 1 to 140 million. So while the lottery plays with few balls, a plant genome has between 100 million and several billion base pairs. Using Arabidopsis as an example, we found out that even if you would generate a new plant every second since the Big Bang, it would still be unlikely to get mutations at 20 desired sites in the genome. So consequently, this threshold criterion is based on a statistically untenable equation, So the proposed approach would go beyond any relevant timescales of breeding and species evolution. On top, the biological parameters make it more complex. So while statistical approaches assume an even distribution, mutations are not evenly distributed throughout the genome. So the mutational bias is, for example, observable between coding and non-coding regions, where the intergenic space shows way more genetic variation. The appearance of multiple mutations in the same gene or between linked gene groups is comparably lower. In addition, conventional selective breeding relies on a recombination to combine desired and separate unwanted mutations. So during recombination, pieces of DNA are broken and recombined to produce new combinations of alleles. However, this recombination occurs more likely between distant regions, and recombination within the same gene or among genes in close proximity is rare. So in summary, general mutation rates do not capture relevant timescale, genetic bias, and linkage drag. So consequently, the basic assumption of the proposal underlies a misconception that all conceivable genetic changes could occur at some point if crossing or mutagenesis will proceed long enough. So it is not the number of mutations, but their constellation that is decisive. And a pure threshold value does not consider this genetic context. The second threshold criterion enables the substitution and insertions of no more than 20 nucleotides. The frequency and type and length of mutations may vary depending on the source of DNA damage, DNA repair mechanisms, the plant type, and the plant genome size. But in its related technical paper, it's described that the criterion is based on data detecting random sequence insertions and substitutions, typically of a length less than 10 nucleotides. The reasoning for a concrete threshold of 20 is the theoretical probability that a random sequence, smaller than 21 nucleotides, may already occur elsewhere in the genome. So also here, there are three other aspects that I would like to address on the next slides: randomness, probability, and design. Although the word random is used several times in the scientific reasoning for the proposal, it is neither defined in more detail nor included in the final criterion. So here is a general definition. Random means lacking any definite pattern, plan, or purpose, something that is not predictable or determined in advance. So we could change this definition. The transition of the word plan can look random and destroy a sentence meaning, or it can grammatically fit and slightly change the sentence. So the argument that a random sequence may already occur elsewhere has no relation to whether it appears random or fits into a pattern at another position. And again, I would like to guide you through the statistical probability using a simplified lottery example. So you remember that the chance to pick the right numbers in the lottery is 1 to 140 million. If the selected numbers should not be drawn in random order, but in ascending order, the likelihood drops further to 1 to 100 billion. So for statistics, the principle of randomness makes a significant impact. With regard to mutations, it matters statistically if 1 or 20 nucleotides mutate into any other nucleotides or into a very specific sequence of nucleotides. So consequently, also this threshold criterion represents a misinterpretation of any realistic breeding scales. The proposal requests only that a targeted mutagenesis technique is used that has been developed since the implementation of the GMO regulation in 2001. There is no further technical distinction. Most established is the original CRISPR-Cas approach, which is also known as site-directed nucleases 1. So here the DNA double strand is cut at targeted sites in the genome to promote an error-prone DNA repair mechanism that leaves small random nucleotide insertions, substitutions, and deletions. Here you see a typical example of a random 1 base pair insertion. It's close to the translational start site, which promotes a frame shift in the open reading frame, and consequently a loss of function knockout mutation. A newer CRISPR-Cas approach, which is known as prime editing, enables the insertion of short specific sequences while bypassing the error-prone DNA machinery. So here you see an example of the precise insertion of 18 nucleotides, which fit exactly into the reading frame of a protein of interest. So these encode a polyhistidine tag that can be used for protein purification or labeling, and it does not affect protein function. So these two examples demonstrate the technical differences between a rough disruption of genes and the precise genome editing to promote a concrete function. In summary, the principle of randomness, the statistical likelihood, and the technical design capabilities have not been considered when drafting Annex 1. So while conventional breeding relies on the unpredictable progression of incremental mutations coupled to selection, NGT interventions are intended to be minimal invasive with a maximum efficiency for a direct functionality. So a pure threshold criterion makes no differentiation in this functionality. Some genetic modifications may be highly likely to occur eventually through breeding. For example, single nucleotide polymorphisms. However, as I showed you so far, there could be also changes that could go beyond what is biologically possible, especially if the two threshold criteria are combined and used to their maximum. In our policy brief, we have compiled 3 examples for proteins, promoters, and non-coding RNAs. Today, I will show you the protein example. Importantly, the 2 threshold criteria do not contain any distance regulations or genetic context restrictions. So up to 20 small insertions could be placed close to each other. So let's say with only a distance of 1 endogenous base pair as a spacer. In protein coding genes, we need to think in triplets coding for amino acids. So 3, 6, or even 18 amino acid nucleotides could be inserted into an open reading frame of a protein coding gene without disrupting the translational reading frame. These are here marked in red. So this could be done up to 20 times in a single protein of interest. And following this 20 times 20 approach, we are not talking anymore about short insertions of up to 20 random nucleotides, but we are talking about up to 400 nucleotides that can be functionally coordinated. And thereby, protein functions could be completely remodeled, or a de novo protein could be designed. While the protein example was humanly conceived, the future will show how AI applications could use the scope of Annex 1. AI has become a transformative, rapidly evolving tool also in the field of genetic engineering, as we have already heard today in the other presentations. So the question whether NGTs only accelerate the breeding of conventional plants within the scope of existing breeding knowledge falls short for AI. AI-driven tools can analyze vast datasets of plant genomes and phenotypes and thereby identifying new targets for engineering. So this includes AI-assisted design of novel genes and proteins, predictions of genetic circuits, and the optimization of metabolic pathways. So looking specifically at de novo design, AI thinks outside the box to expand the evolutionary limited protein sequence space. As a consequence, also small insertions could in constellation exhibit characteristics of intragenesis, transgenesis, or even synthetic biology. Importantly, the proposal stipulates that NGT1 plants that are crossed to each other remain NGT1 plants. So, crossbreeding could produce NGT1 plants with multiple de novo proteins. In theory, the combination of multiple de novo proteins could even function synergistically or as a signaling cascade to elicit AI design traits. So this increasing convergence between AI, NGTs, and synthetic biology has not been considered in the proposal. The AI-assisted design or the novelty of a molecule or a system is not an exclusion criterion. So mere threshold criteria do not capture the underlying complexity and the impact of AI. The proposal tolerates risks of NGT1 plants by comparing them with risks posed by conventionally bred plants, which is considered within the scope of proportionality. So the assumption of equivalence thereby conveys an unjustified sense of safety, as risks are not excluded per se. The risk potential of any plant is not linked to the type, size, or extent of a genetic modification, but it unfolds through genetic context, functionality, phenotypic expression, and the interacting environment. So all these aspects have not been addressed in the proposal, but they require a case-by-case risk assessment. Compared to conventionally bred plants, NGTs enable a greater depth of intervention and the specific orchestration of previously unattainable genetic modifications. With AI-assisted design on top, there is the possibility that there is no evolutionary comparator in a species and that the de novo designed molecules or systems appear new to nature. And accordingly, such novel NGT plants might pose similar or greater risks than GM plants produced and risk assessed so far. In future, genetic modifications could be developed specifically to meet the Annex 1 criteria for the NGT1 fast track, which would also bypass the comprehensive risk assessment. So in summary, neither the proposal's overall concept nor the threshold criteria are able to properly consider the intended and the unintended risk-related effects of NGT plants. Coming to the end of my presentation, I would like to conclude the following. The proposal's concept does not consider the statistical probability within relevant breeding scales, the genetic bias, context and functionality, the technical range of NGT applications, the convergence of NGTs and AI, the intended and unintended effects of the phenotype, and the risk-related outcomes. So therefore, our analysis reveals that the proposed assumption of equivalence in its current form is statistically, scientifically not justified and not suitable for regulatory purposes. There are further considerations concerning Annex 1. So first, the possibility to include also the other Annex 1 criteria broadens the spectrum of genetic modifications, the AI design space, and potential risks. And also, the regulatory process is still ongoing. So the negotiations in the trilogue between Commission, Parliament, and the Council could change the Annex 1 criteria. In order to meet the original objective of the proposal and to ensure a high level of protection of human and animal health and the environment, the genetic context, the impact of AI, and possible risks must be considered. I thank all of you for your attention, and many thanks go to the organizers of this workshop for giving me the opportunity to present our policy brief. And if you would like more details, you can find the related publication with this QR code. And now I'm looking forward to your questions and to your comments. Thank you very much.
Thank you so much, Juliane. So I think a lot of scientists would agree that the Commission's criteria are unscientific, but it's very useful to have you breaking it down in detail for us. In the political debate, there are a lot of people who are trying to defend the criteria that the Commission has proposed, but if you look closely enough, then you will find that even the Commission fails to provide scientific evidence for their criteria. I also want to remind that this criticism has been raised several times already by the Federal Agency for Nature Conservation, but also by the Austrian Federal Environment Agency and by ANSES, which is the French Food Safety Authority. So there are quite a lot of authorities and scientific bodies that raise similar concerns. I think it's also a question on a more philosophical level of who we want to be as Europeans today. In the times we are living, with so much attack going on science and scientists, I believe we should really take pride in basing our public policies on solid scientific evidence and not some magic numbers. The debate is not over yet. As we said earlier, trilogue negotiations continue, so there is still room for improvement. And I think there should also be room to think about, keeping the current legislation, which is actually a much better regulation than what has been proposed now by the Commission. So by adapting it a bit to new technological developments, it could serve us quite well. With this, I would like to thank you for listening today, and I hope we will have more occasions to continue the discussion. Thanks for our speakers, thanks to the interpreters, and hope to see you all soon again.